Hi, my name is Kendra Lilly. I'm in the 8th grade. I go to Southeastern Junior Senior High School, and this is my second year in Science Club.
Hi, my name is Zoey Griswold. I'm an 8th grader at Southeastern Junior Senior High, and this is my first year in Science Club. Our project is Water Warriors using solar disinfection to clean contaminated water.
The purpose of our project is to see if solar disinfection can clean contaminated water from an untreated source. Our untreated source is the Mississippi River. The samples we use will be exposed to a full spectrum of light, including UV rays, and they will be put under the light for a various amount of time.
Based on research of solar disinfection, we predicted that there would be an indirect correlation between the time of full spectrum light exposure and the amount of bacteria growth. We hypothesized that as the light exposure for the Mississippi River water samples increased, the percent of bacteria growth would decrease and vice versa. Furthermore, we predicted that the untreated samples without solar disinfection would have the most bacteria growth on average.
To start our project, we labeled 8 plastic bottles for 6 hours, 12 hours, and 24 hours, and 1 without light. One set was for Mississippi River water, and the other set was for spring water. We then collected the water from the Mississippi River and labeled 8 petri dishes. We filled the petri dishes with 20 milliliters of nutrient agar, and we allowed them to cool and solidify. We set up 3 solar disinfection stations using clamp lamps, 100-watt reptile bulbs, light timers, and ring stands. We then set up 1 station for without light. We added 355 milliliters of Mississippi River water to the 4 bottles and 355 milliliters of spring water to the 4 bottles. We laid the bottles horizontally below the lamps in the corresponding stations on a dark metal sheet. We set the timers and left the bottles under the light for the corresponding amount of time. We dipped sterile swabs in the water and spread the water sample over a plate with even distribution. After 24 hours, 48 hours, and 96 hours, we measured the amount of bacteria growth on each plate. We placed a transparent grid over the plate and shaded the squares where the bacteria was present. We then calculated the percent of bacteria covered on the surface and recorded the results. We used these procedures and conducted the experiment 4 more times. We then averaged the graph results using Microsoft Excel and calculated standard deviation.
Our independent variable, which was the one thing changed, was solar disinfection time. We had 4 different stations set up, and we had 1 which was control, which was no solar disinfection, then 6 hours, then 12 hours, and 24 hours. Our dependent variable, which was the thing that was measured, and we measured the percent of bacteria growth. Then our control variables, which were the things kept consistent, were our testing environment, which was in the science lab, our water sample source, which was the Mississippi River water, and the type of light, which was 100-watt reptile bulbs, and the measurement technique, which was we used this transparent grid and we put it up to the petri dish and we shaded the squares that had bacteria present.
As we expected, spring water had no bacteria, so we did not graph it. This graph shows the amount of bacteria growth at a 24-hour reading, 48-hour reading, and 96-hour reading. The y-axis shows the percent of bacteria growth, and the x-axis shows the reading times. At 24 hours, there was no bacteria growth. At 48 hours and 96 hours, the plates with river water with no solar disinfection time had 100% coverage. As the light exposure increased, the bacteria growth decreased. At 96 hours for the 24-hour solar disinfection time, it had significantly less bacteria growth than the 6 hours and 12 hours because the air bars do not overlap.
Based on research of solar disinfection, we predicted that there would be an indirect correlation between the time of full spectrum light exposure and the amount of bacteria present. We hypothesized that as the time of light exposure for the Mississippi River water samples increased, the percent of bacteria would decrease and vice versa. After conducting our 5 trials and calculating the average standard deviation, we determined that our hypothesis was correct.
Great effort was made to conduct this project in a scientific manner. However, some variation occurred in the results. A technique was established to distribute the samples on agar plates but there could have been slight differences in the sample taken and the coverage. Thank you for listening.